So, we are now going to discuss about the initiation of replication in eukaryotes. Normally, last we have done about the replication in the lagging strand in eukaryotes. Here we will discuss about the initiation process, that how eukaryotic replication begins, and on the same time I will also try to discuss about the problems that occurs during the end replication. Okay, so initiation of replication in eukaryotic cells requires two steps to occur at distinct times in the cell cycle. So there are two steps which has to be done for initiation of replication in eukaryotes, and the two steps are replicator selection, as given here, replicator selection and origin activation. What is replicator selection? Replicator selection is the process of identifying those sequence that will direct your initiation of replication process. So it is a process of identifying those sequence, which sequence will be the initiation sites in eukaryotes, and this event occurs in the G1 phase of your cell cycle. This process will leads to the assembly of multi-protein complex at each replicator in the genome. So the first step for initiation of replication in eukaryotic cells is your replicator selection. So first, the proteins which are involved for initiation, they will identify the sequences that will direct your initiation of replication process, and this replicator selection, that which sequence will be your initiation sites, this event will occur on the G1 phase of your cell cycle, and this process will leads to the assembly of a multi-protein complex at each replicator in the genome. So assembly of multi-protein complex, many proteins will be associating with that particular region that we are calling as replicator in the genome. Now eukaryotes have multiple origin of replication, as we know, because eukaryotic genome are pretty large than the prokaryotes. So we have to establish the whole copy of the DNA. Okay, so that it can divide into two daughter, so that we can distribute them into two daughter cells during cell division. For that, we need quickly access to this replication process, and thus they have multiple origin of replication, which we are calling as replicator here. And for identifying that which sequence will be the replicator, for that this step is crucial, and multi-protein complex will associate with these replicator regions in eukaryotic genome. The next step is your origin activation. Origin activation occurs only after the cells will enter to the S phase of your cell cycle, and it will trigger the replicator associated protein complex to initiate the DNA winding, unwinding, and DNA polymerase recruitment. So origin activation is the step where the replicator associated protein complex, which was associating with the replicator regions, they will now starts to do their function. Okay, by binding with the replicator region does not means that they will function. It functions only after the cells have reached to the S phase of cell cycle. It is catalyzed by some sort of protein kinase that you will see later on, and during this time of origin activation, at that time only your replicator associated proteins will start to function. They start to initiate the DNA unwinding, and they will also recruit the DNA polymerase. Okay, the three types of DNA polymerase that normally works here, like your DNA polymerase alpha primase complex, which initiates your primer formation, then the polymerase delta and polymerase epsilon. Replicator selection is actually mediated by formation of pre-replicative complex. So for the first step that we have talked about the replicator selection, some sort of proteins must mediating this function that are replicative complexes. Okay, uh, the pre-replicative complexes are commonly called as pre-RCs. As you can see here, pre-RCs. This pre-replicative complex is made up of 4 separate proteins. So this pre-replicative complex, directly you can say this pre-replicative complex is actually identifying that replicator regions and bind, and it is made up of 4 separate types of proteins that gets assembled in an ordered fashion at each replicator sequence. Let us see the name of that protein. The first step is in the formation of pre-replicator complex is your recognition of the replicator by the eukaryotic initiator ORC. So the first protein which binds to that replicator region, okay, that protein name is ORC. ORC. Now once ORC protein bound, it will recruit 2 helicase loading proteins known as CDC6 and CDT1. So once ORC bound, it then recruits 2 other helicase loading proteins known as CDC6 and CDT1. So these are the 2 proteins. CDC6 and CDT1 are helicase loading proteins. They will allow now the helicase to bind together. First ORC had been bind with the replicator region by identifying that which is the replicator region. Then it will recruit the 2 helicase loading proteins, that is CDC6 and CDT1. Together ORC and the 2 loading proteins will then recruit a protein that is thought to be eukaryotic replication fork helicase. Okay, the name of that helicase here is MCM 227 complex. Okay, MCM 227 complex. MCM is mini chromosome maintenance. Okay, and this MCM is thought to be function like helicase, just like in prokaryotes. Okay, so this is the what can I say, this is the pre-replication complex which actually identify the replicator region. Okay, and they bind in an ordered fashion. First ORC bind, then ORC will recruit the helicase loading protein, that is CDC6 and CDT1, and then they are recruiting the MCM 227 complex, which is a helicase. Now formation of this pre-replication complex does not lead to immediate unwinding of the origin DNA. Okay, by binding of this pre-replicative complex, it does not immediately starts to function. Instead, the pre-replicative complex that are formed during G1 are only get activated to initiate the replication after the cells have been passed from G1 to the S phase of the cell cycle. So simply, this pre-replicative complex, after binding to the replicator region, which are the origins of replication, after binding they will not function. They will function only after the cell has been goes from G1 to S phase. Okay, let us see the diagram now about this pre-replication complex. Okay, so this is the diagram of pre-replication complex. Here one replicator region has been shown. The ORC, the first protein that assemble in that replicator region. ORC then recruits the two other helicase loading proteins, that is CDC6 and CDT1. Then these helicase loading proteins now allows the MCM227 complex to bind. MCM complex is thought to have a structure and function is similar with helicase. So in this way your initiation of your replication in eukaryotes begin. It is given on figure 8.29 in your Watson book. Okay, so let us move forward. So it is saying that this pre-replicative complex can only get function after the cell have reached to the S phase of your cell cycle. But how? There must be some proteins which are regulating this function. These pre-replicative complex are activated to initiate replication by two protein kinases. So there are two protein kinases which are regulating the function of pre-replicative complex. And what are those name of these protein kinase? CDK and DDK kinase proteins. So the pre-replicative complex are activated to initiate replication by two protein kinase which are CDK and DDK kinase protein. Kinases are normally the proteins that covalently attach the phosphate groups to the target proteins. What are kinases? Kinases are those proteins that covalently attach phosphate groups to the target proteins at specific amino acid residue of your target proteins. Obviously, there are some specific amino acid of the target protein. In that amino acid, they try to add the phosphate groups to make them active. So the CDK and DDK kinase proteins are needed here for activating the pre-replicative complex to function. Each of these kinases are inactive in the G1 phase and is activated only when the cells enters to the S phase. So each of these kinases, whether it is CDK or DDK, they are inactive in the G1 phase and it gets activated only when the cells have entered to the S phase, right? And once activated, this kinase will target the pre-replicative complex. It will try to phosphorylate them and other replication proteins. Phosphorylation of these proteins will result in assembly of some other additional replication proteins at the origin, and then the initiation of the replication begins. So this kinase become activated only when the cells enter to the S phase. Then the kinase will try to phosphorylate the target pre-replication complex and other replication proteins. Phosphorylation of these proteins will result in assembly of some other additional proteins at the origin and the initiation of replication. Now the new proteins that are getting recruited here at the origin are your 3 eukaryotic DNA polymerase, that is your DNA polymerase delta, epsilon and alpha, and the number of other proteins that are needed for initiation, that is your primase, such as primase, which are required for their recruitment. Okay, so these are some additional proteins. After phosphorylation of the pre-replication complex done by the kinase, it will result in assembly of some other additional proteins at the origin and the initiation of replication begins. And what are those additional proteins that are getting recruited now? the three types of DNA polymerases, the DNA polymerase alpha, delta and epsilon, and the number of other proteins like your primase which are needed for synthesis of primer. Okay, interestingly, the three different polymerases assemble at the origin in a particular manner. So if you see the three different polymerases which get assembled here, they are assembled at the origin in a particular manner. Like your DNA polymerase delta and DNA polymerase epsilon associate first, then it is followed by DNA polymerase alpha and primase complex, because our polymerase alpha associate with primase to form that complex. So first DNA polymerase delta and epsilon bind, associate with that region, and then it is followed by DNA polymerase alpha primase complex. This order will ensure that all three DNA polymerase are present at the origin before synthesis of the first RNA primer, which is catalyzed by the pol alpha primase complex. So this type of arrangement normally occurs so that your when synthesis of your DNA begins, they can able to do it in a stepwise manner, like the pol alpha primase complex are observed first, which will synthesize the primer. Okay, and then it is followed by the DNA polymerase delta and epsilon. That's why they have arranged in that way. Only a subset of proteins that assemble at the origin go on to function as a part of eukaryotic replisome. The other proteins, so out of all of these proteins that I have said, the ORC, CDT, CDC1, okay, CDC1, then CDC6, then I have said the MCM227 complex, the three type of DNA polymerase, other proteins which are recruited. So some of them, only a subset of these proteins will now assemble at the origin and go on to function in the elongation step as a part of eukaryotic replisome. The other proteins like your CDC6, like CDT1, which are the helicase loader proteins, they are released or they are getting degraded after their role is complete. So now as they have finished your initiation process, they will now go for elongation. For that, those proteins which are needed for elongation, they will remain in the eukaryotic replisome. The other proteins which are no longer needed, like for example CDC6 and CDT1, which are helicase loading proteins, they will get degraded or they will get unwinded or released. Let us see in the figure so that it will be easy to remember. Okay, so here we can see the DDK and CDK. They are actually the, what can I say, they are actually the kinase which triggers activation of these protein complexes, the pre-RC, pre-replicative complex. The other auxiliary factors and proteins are getting recruited, like you can see the delta, epsilon, some other factors are getting binded, whereas the other proteins which are no longer needed like CDC6, CDT1, which are the helicase loading proteins, they get disassociated from the pre-replicative complex. And now you can see they have opened, unwinded the region, they have started to function. The helicase is moving in a direction of replication fork, as you can see, in both direction, because bidirectional replication normally occurs in eukaryotes. And then you can see the polymerase delta epsilon is carrying on to position in order to synthesize polymerase alpha primase complex, then first comes, then associate, then and synthesize a short RNA primer, RNA DNA primer. As you can see, a short RNA primer is followed by a synthesis of short fragment of DNA, as we have studied in the lagging strand synthesis, that the Pol alpha primase complex synthesizes short RNA DNA primer. Sliding clamp and clamp loader then get recruited here. The Pol alpha primase complex get released. Okay, so as the elongation process proceeds, then most of the proteins get dissociated. Now the Pol alpha primase complex function is over, but its function is not over in the lagging strand, because repeatedly this Pol alpha primase complex will be required in the lagging strand. Okay, and that's the reason that Okazaki fragments occur in the lagging strand, where discontinuous DNA synthesis occurs. And start lagging strand synthesis, you can see lagging strand synthesis is being occurred. Okay, so this is your lagging strand synthesis, you can see, whereas the leading strand synthesis is quite smoothly it is occurring. Okay, so in this way your synthesis of eukaryote replication begins. Let us move forward now. Okay, so now we will discuss about, this was about the starting of the initiation and how it elongates. Okay, there is not much in detail about the elongation. The elongation is pretty straightforward. Leading strand will have a continuous DNA synthesis, whereas lagging strand will have a discontinuous DNA synthesis. The problem that will be occur is during the end of replication. What problem? Let us see. It is saying that lagging strand synthesis is unable to copy the extreme ends of linear chromosome. Okay, so the lagging strand synthesis will be unable to copy your extreme ends, that is extreme 3' or 5' ends, where the lagging strand synthesis is occurring, will be unable to copy that region, and that's why a gap will be created. That part will not be replicated. So let us see what they are trying to say. The requirement for an RNA primer to initiate all new DNA synthesis will create a problem for the replication of the ends of linear chromosome. The requirement for an RNA primer, so we always need an RNA primer here to initiate your new DNA synthesis, and that's where it creates a problem for the replication of the ends of the linear chromosome, because the ends of the linear chromosome are not replicated at all. That is called as end replication problem. How it occurs, let us see. The end replication problem occurs especially in the lagging strand. How? The requirement of many multiple primers to complete the lagging strand synthesis means that a complete copy of its template cannot be made. Again I repeat, the requirement of many multiple primer in order to synthesize the lagging strand means that a complete copy of its template will not be made. Complete copy of its template cannot be made. Why? As the lagging strand replication machinery reaches to the end of the chromosome, at some point what will happen? Primase could no longer has sufficient space to synthesize a new RNA primer. There will be a small gap. Within that gap, how will primase will able to synthesize primer, or it will able to fulfill that gap by DNA polymerase? Or even if the primer is added, suppose the primer has been added somehow, in order to fulfill the replication process, in order to complete the replication process, we have to remove the RNA primer. So if the last RNA primer for that Okazaki fragment is removed, there will be what? There will be a short gap, there will be a short region of unreplicated single-stranded DNA at the end of the chromosome. So that's the in replication problem. First, it is saying that as the lagging strand synthesis continues, more and more RNA primer are getting recruited for initiation of the replication in lagging strand. And what happened at the end of the replication, what happened, there will be a very less space to synthesize a new RNA primer, and thus the gap will be created. Or even suppose a new RNA primer has been recruited somehow at the end of the of your replication, at the end of your strand. Okay, then also what happened, as a rule, when you are completing the replication, you have to remove that last RNA primer, and by removing the last RNA primer, there will be remaining a short region of unreplicated single-stranded DNA gap at the end of your chromosome. So this means, so this small gap means, oh, it's just nothing, but these small gaps will be huge, because at each round of DNA replication, it would result in shortening of one of the two daughter DNA molecules. Each time when you are doing replication, the lagging strand will be becoming shorter, shorter and shorter, and eventually the genes, the essential genes that are found at the end of the chromosome, will be getting lost forever. Okay, so how we can solve this problem? It can be solved in various ways normally. Like here, two ways has been given, two important ways that are normally discussed, and researches is also going on nowadays. So one solution is we can use a protein instead of RNA as a primer for the last Okazaki fragment to be replicated. Okay, so the first solution is we can use a protein instead of using an RNA as a primer for the last Okazaki fragment at each end of your chromosome. In this situation, what happened, the priming protein that will bind to the lagging strand, they will use an amino acid to provide that OH group, and with the help of that OH group, the DNA polymerase will able to synthesize the nucleotides. This is the first way, okay, to extend the replication process. So the first way that we can solve that end replication problem, a gap is created at the end of your chromosome, is by recruiting a protein instead of RNA primer for the last Okazaki fragment synthesis. Now in this situation, that protein will bind to the lagging strand template, and they will use an amino acid to provide that OH group, and you know that DNA polymerase can only add new nucleotides to a 3' having the OH group. So by providing that OH group by that protein, maybe that DNA polymerase will able to synthesize nucleotide based on this, and we can extend that replication process. This type of process generally occurs on some species of bacteria and viruses that have linear chromosomes. Generally bacteria have circular chromosomes, but those some species of bacteria having linear chromosomes, they do this type of process. Okay, let us see the picture now. So here you can see a protein, DNA polymerase OH, they are synthesizing, okay, and they are solving the end replication problem as stated in your notes. Okay, so in a well way we can see the protein has been bound to the end, and they try to, with the help of the OH group, DNA polymers will bind and able to synthesize the nucleotides, and there is no problem of the end replication. the end part of your replication fork will be completely be synthesized. Okay, this is given in the figure 8.35 in your Watson book. Okay, what is the another way? The another way that is done is your telomerase. You have heard about the chromosomes, you have heard about the chromosome structure, that the end of the chromosomes have telomerase, right? Sorry, not telomerase, telomer, Telomere, and the enzyme that catalyzes the formation of telomer is telomerase. So most eukaryotic cell uses entirely a different solution to replicate their chromosome ends. Okay, so how do they end that problem, that end replication problem? So how do they do that? So the ends of the eukaryotic chromosomes are called telomers. The ends of the eukaryotic chromosomes are called as telomers. Telomers are actually Tg rich DNA sequence. The telomers are actually Tg rich sequences that are replicated tandemly, means repeatedly. So telomers are those regions which are very rich in Tg and they are repeated tandemly, means one after another. Suppose for example in humans the telomer sequence is double T A triple G, 5 prime double T A triple G, double T A triple G. Okay, so by recruiting this telomer sequence at the end of your at the end of your chromosome, so this unique structure acts as a novel origin of replication that will help you to compensate. Okay, compensate means the loss that occurs during your end replication as a result of end replication problem. It will fulfill that problem, it will try to overcome that mistake by solve that mistake by adding this telomere sequence. Okay, and on the based on the telomere sequence, finally the DNA polymers will able to synthesize the last nucleotides on the end of your replication. Okay, on the end of your chromosome. So this unique sequence will act as a novel origin of replication that will help you to solve that mistake, that is compensate for the end replication problem. And this unique structure, that is the telomere sequence, is carried out by a special type of DNA polymerase enzyme known as telomerase. So in eukaryotes, how do they solve that end replication problem? By synthesizing repeated sequence, tandem repeated sequence called telomers. Telomers are Tg rich DNA sequence which are repeated tandemly one after another. Like in humans, the telomers are repeated in this way, double T A triple G, double T A triple G. Now this unique structure acts as a novel origin of replication. Okay, so that your DNA polymerase can able to bind and can able to fill that gap that occurs at the end of your end of your chromosome. Okay, end of your DNA. Okay, and thus in this way the telomere sequence are actually overcoming that mistake, are able to solving that mistake for the end replication problem. Now who catalyzes this telomere sequence? Must be there is some enzyme, and for that the enzyme that catalyzes this formation of telomere is your telomerase. Okay, what is telomerase? Let us see. Telomerase is an RNA containing enzyme. It has both protein and RNA components. So in short you can say it is a ribonucleoprotein. Okay, it has RNA as well as it has protein. With the help of that RNA they are synthesizing the nucleotides. Okay, this repeated sequence double T A triple G in case of humans, human telomere. It does not need an exogenous DNA template to direct the addition of new nucleotides. So this telomerase who is that is catalyzing the formation of telomere sequence, they do not need any template because they have their own RNA, and on the basis of that RNA they use that RNA as a template to synthesize the strand. So instead, the RNA component that is found within the telomere, the RNA component that is found within the telomerase, they are serving as a template for adding the telomeric sequence to the 3' end at the end of the chromosome. Okay, so this is the function of telomerase. Okay, if you look at the structure of telomerase, it is given differently in different books. I will show you the structure also. So it is an RNA containing enzyme. That means it is a ribonucleoprotein. It has an RNA as well as protein, and it does not need any DNA template to synthesize DNA. Instead, how it do? As they have an RNA, these telomeres have an RNA component. They use that RNA component as a template, and they use that RNA as a template for synthesizing addition of the telomeric sequence to the 3' end at the end of the chromosome. Now telomerase are specifically elongate your 3' end OH of the particular single stranded DNA. Telomerase specifically, they are elongating the DNA to the 3' end. The gap is still remaining, the end replication gap still remaining, but telomerase is elongating the 3' OH of your particular single stranded DNA using its own RNA as a template. So the newly synthesized DNA is now single strand. Now when telomerase is acting on 3' end of your telomere, it only extends the 3' end of your chromosome. So how the 5', the gap that was already present, how this 5' gap can be filled? Now to accomplish this, again your lagging strand DNA machinery will come and synthesize the last fragment. Okay, fill that gap, and thus your end replication problem will be solved. So what it does, telomerase is synthesizing the telomere repeated sequence at the 3' end. The 5' end has a gap. Now the DNA polymerase enzyme, that means the enzyme which are used for lagging strand synthesis here, they will now observe the 3' OH. So they will try to bind there and fill that gap. Okay, fill that gap in the 5' end. Okay, and in this way, by providing an extra 3' end, by providing an extended 3' end, telomere provides additional template for action of the lagging strand. So telomerase is synthesizing the telomere sequence, right? And by synthesizing this telomere sequence, what happened? They are now using as a template for the lagging strand replication machinery, which can then extend the 5' end of your DNA. Now let us see the figure finally. So here it is the figure. You can see a telomerase protein. It has been shown in this way. It is a ribonucleoprotein showing the 3' and 5', and an RNA. It is used as a template. It also has an RNA, and it with the help of this RNA as a template, it tries to synthesize that single strand towards the 3' end. Okay, in this way they are translocating. That means they are moving and try to synthesize as long as of the DNA towards the 3' end. Okay, now how the gap is filled? So here you can see the telomerase extends the 3' end of your telomere, where you can see above the gap is still there. Addition of 3' end DNA now act as a template for a new Okazaki fragment. So you have to see it carefully here. You can see the 3' end is extended by your telomerase enzyme catalyzing the synthesis of telomere. The repeated sequence double T A triple G like in human telomere, and now as they have extended quite long, so that gap is now quite big. So now we can use this telomere sequence as a template for allowing the lagging strand machinery to come and synthesize the last Okazaki fragment. Okay, so additional 3' end of the DNA can act as a template. The additional 3' end DNA can act as a template for new Okazaki fragment. So here we can see, and we fulfill that gap. Okay, this telomerase extension still has 3' overhang, but it is of no use because it is a repeated sequence. Okay, and in this way the extension of the 3' end of the telomere by telomerase will solve the end replication problem. But what happens as the cell division approaches more and more, these telomeres get shortened after quite cell divisions, and thus that leads to telomere shortening, and then again your end replication problem begins. This is the main reason that leads to aging. Researches are going on based on this particular topic, that about the telomerase with the aging process, that telomerase is solving the end replication problem one time. Okay, but after considerable cell division is occurring, more and more the lagging strand is getting shortened. Okay, but after considerable period of time there will be a one time when again your essential regions of your gene will be getting eliminated due to problems occurred in at the end of the lagging strand. Okay, so this was all about the telomerase replication, then about the initiation of replication in eukaryotes. Okay,